Oh, yeah.
That looks like one of those coral predators.
Sneaking up.
Very unsuspecting predator.
Yeah.
So there was I had a conversation lunch today with other Steve, who was on, I don't know which watch, is he 12 to 4 or 48?
He's 12 to 4.
Yeah, 12 to 4.
Yeah, and he was saying, I was saying yesterday we were seeing in the areas of high density what looked like some kind of structural, like globular sediment that was drifting off of Herc. And everyone was telling me, oh, it's coming off of Herc, it's coming off of Herc. But they saw this too, and they thought it might be larvae. And I was starting to think during that portion when we were close to those corals, maybe it might be larvae, because they were slightly pigmented, like pink.
Yeah, they look pink.
Yeah.
Little rounded pink balls.
And
like
so
cherry blossoms,
maybe
There's a holothurian there.
Oh, yeah.
Is it possible we saw a spawning event?
I don't know. It could just be that maybe the thrusters suspended some.
Well, we
some
did see that those
particular-
black corals, for instance, they looked.
Gravid?
Yeah,
Yeah.
I was wondering what that could be, but anyway, that's a bit of a tangent. I was kind of going to remark on the turbidity of the water column,
Mm-hmm.
but kind of got sidetracked with that thought in my head.
Walteria off to the right. Video, are the lights for Herc up all the way?
We have also the uppermost lights are decommissioned right now.
Oh, okay.
So it might be a little bit dark for
I
that
see.
reason as well.
Thanks for clarifying.
Yeah, come up there, Jake.
Oh, wow. Look at the view in Argus of those large sponges to our left.
yeah.
So as we're rising, we're definitely
yeah.
getting into Poliopogon territory. We want to zoom in on that sponge?
Looks like it's real.
Which one?
Like pipe sponge.
Sure thing. Reg.
Yep.
Cool feature then.
Yeah, for sure. That ship move is complete. Argus has another 10 meters to go. We can, back row, you want me to call in another one, continuous, or?
Since we didn't
Gonna
see
hang
another
around.
one of those Acanthogorgia, maybe we keep moving?
Oh yeah, I think.
Yeah, okay. Yeah, go ahead and call in another one, Rennie.
Sure
Push on in there, please.
I don't know, even in the couple hundred meters we're moving across the vertical today, I don't think we'll really move out of the range of any of these species.
Okay. Thanks, Jess and video. That's good.
Sure thing.
There's a lot of Calyptrophora here as well. The pink coral on the right, almost certainly.
Mm-hmm.
Could be Narilla, but has a very lyrate branching, which is usually indicative of Calyptrophora, Paracalyptrophora first.
So this is the first kind of pile of sediment we're seeing, and interestingly, it's like on a very steep wall.
No ripples either.
No, I'm not seeing any ripple features. There's another holothurian off to the top left.
It's a big one.
Nice big purple one.
Purple sea cucumber.
Oh.
Color is unreal.
Yeah,
Wow, yeah.
very beautiful.
It's like fluorescent highlight.
Like some
highlighter
of our
color.
nice sunsets out here,
Yeah.
that color we get.
Do you guys want to zoom on it, or did you?
Maybe just a snap zoom, it's
Good
fine.
partial there. Sorry.
Pew, pew. Cucumber. What are
Matches our lasers very nicely. Compliments, rather.
Great.
Yes,
Income
great.
Sorry, we're
Looks like a potato.
I would not eat that potato.
Purple potatoes are good, but maybe
Yeah,
not.
purple potatoes
Maybe
are really
not
good.
ones that are that shade of purple.
Might be a little
There is something called a sea potato.
Yeah.
What is that?
I think it's a type of urchin, actually.
Huh.
Interesting. Jake, you want to risk? Do you want to make sure Mon goes all the way to stowed to the left there? Feels like it's in the frame pretty well.
Some cool looking sponges off to the right.
You're tilted down a
There's
little bit, aren't
there's
you?
a whole line of sea foods.
Sea strawberries.
Yeah, remember that from the west coast, the sea pickles?
Oh yeah, those are very cool.
Oh yeah, the pyrosomes.
Yeah.
Those things are sometimes very prominent in the first few 100 meters.
That was always very interesting, yeah.
Yeah, whether it was going to be a sea pickle recovery or not.
Exactly. When you hit them, then they start to light up.
Right.
Bounce off the ship.
What are the rock core sampler requirements today? Are we still looking for angular?
Yeah, so there's definitely an interest in trying to get rocks that are worthy of trying to get dating material here.
Are we going to sample those, or are we going to wait for the next watch?
If I see something that strikes me as being good, what we're looking at right here looks really altered. So I haven't seen something that really is jumping out to me as a good candidate. But if I see something, I'll definitely try to get it. Otherwise, I'm sure Andrea is interested to go rock hopping.
Would you say it's highly altered?
Good.
It is highly altered.
Oh no, down we go. Oh wow, keeps going.
That actually looks kind of fun, right?
Yeah.
Jeff, you got the Wilhelm screen over there?
Yes, I did. I just had my mic on.
Can we please cut that into a highlight reel?
Sorry, viewers, if you've been submitting comments for the last 4 or 5 minutes, they're not coming through, but I will try to get to them as soon as we reconnect.
Not a bamboo coral in sight. That's surprising to me.
I don't remember from Seamount A, south of here, did we see bamboo coral on Seamount A?
It's all fuzzy.
Yeah, I know. That was like 13 dives ago.
Yeah, I can check the report, but I think we saw some of the big, thick, unbranched ones.
Okay.
It's all the tall ones.
Yeah.
Right, because the highlight video was from Seamount A.
Okay.
Unbranched bamboo coral.
I haven't seen the highlight video yet.
Yeah.
All right. So it looks like maybe we crested a very small knoll or maybe coming down into a slight saddle, and then it'll start going back up the ridge again.
There's another one of those really nice crinoids there.
Where is that?
On the sponge there.
Oh yeah, on the Walteria,
Alteria.
if that is indeed what it is.
Yeah, we can get a partial zoom on that.
Oh, I see which one, the dark.
Yeah, the
Purple
purple
one.
one.
I was looking at those upper orange ones.
There are a lot of corals here.
push
If you
on in
look
there,
close
please.
enough, they're just smaller colonies.
Yeah, I just was noticing that one off to the left.
Very cool.
There's after we do this, there's a cup coral to the upper left-hand corner that I wanted to check out.
Okay. There's another, like the stick off to the left too.
Maybe Sticopathes.
Sticopathes.
Is this Walteria more pink than some of the others we've seen?
Sometimes they can have a little bit of color to them, but it could just be the refraction of the color from behind.
Thanks, video. We can
Okay.
pull out a little bit. There's a couple other things we want to image here if we can.
Sure.
Where did you see the cup coral?
It's just on the very edge of the frame. Yeah, it's
Right up
pink,
there.
pinkish orange circle. Yeah.
Roger.
I think it's a cup coral, but you never know until you get closer look, because sometimes they look like a cup coral, but they're actually anemones, or anemones can look like
Look
cup
at the
corals.
size of this holdfast over here.
Yeah,
Wow.
Go ahead and push on in there, please, Jeff.
pretty huge. Yeah, so it does look like a cup coral.
This one here.
Yeah.
Yeah.
Any more?
Can we get a little
zoom?
closer? Zoom.
These things are notoriously difficult to ID in situ, but get some imagery nonetheless. But you can see this calcium carbonate sclerosepta, the kind of blades between the tentacles.
Mm-hmm.
Do you see the
Yeah,
there's a
little
polyp
polyp
in the
over here
little
too.
sediment on the left?
Oh yeah, little, what is that?
Solitary hydroid?
Maybe, or solitary, or maybe it could be a settled sea pen.
Oh, yeah.
Yeah. Okay, great. Thank you for
Wide.
the photos. Sometimes sea pens, yeah, they'll form a single polyp that sticks out. Sediment didn't look too deep there, but we know that not all sea pens need very thick sediment.
Catching up on comments and questions here.
Okay. Thanks to everybody that's tuning in, watching the real-time exploration of Ika seamount with us. Last dive of our expedition here. This part of the world. I don't think we've seen any fish yet, have we, on this dive?
I don't think so,
Not a
no.
one.
That was something that struck out to me on our last dive on that ridge feature southeast of Tamana Seamount. So we spotted a number of fish on that dive. I think actually we maybe saw one fish multiple times. So I know that they did a circle around an area. Off in the distance, it looks like there's some rather large sponges in Herc's view.
One of the nice things about working shallower water with ROVs is that the fish are always behind you, and you can always see them with the Argus cam. But sometimes you can see that when we're down this deep, but the fish will always seem to know where the camera is, and they'll always tend to avoid it.
They're like Jeff.
Kind of like people on Nautilus.
Jeff was in front of the camera today.
Yeah, you got me on camera
We
today.
had a great interaction. Cascade Middle School.
Awesome.
You want to go ahead and do a partial
That is
there,
a
Jeff?
fuzzy sponge.
Yeah, a lot of life on that.
Get a better angle here.
I think there's probably more brittle star and crinoid biomass on that sponge than there is sponge.
Well, maybe that's why there's so many broken sponges.
Yeah. That's lovely.
Yeah. You would think if the flow conditions were that good, they wouldn't need to be so high up, but I guess they just...
Maybe it just, like, why not keep going?
Yeah.
Speaking of, do we have an indication yet of which way we think the current is moving here?
Oh, wide there, please. Oh,
Seems like the particles are coming at us kind of from the top right.
kind of from the other right there.
So almost from the southeast.
Yeah.
Okay, a little different. very large sponge here.
Another Poliopogon. It seems to be the sponge of the cruise.
Yeah.
And again, its concave side is kind of angled towards the, let's see, we're looking at it this way. Yeah, it's angled towards the southeast. So is that the predominant direction of current down here? Thanks, Jess.
Yeah, sure thing. Full wide. I'll come back up towards you a bit there.
So we're facing kind of east-northeast. Is there, how much more up is there to this ridge? To the northeast, or is it just all southeast?
The ridge is still on this side of you, right?
Yeah, it looks like it's still, well, that, yeah.
It's not fan. Can we
Might
look
just
to the
be some
left
gains.
of the sponge? To the coral on the left of the sponge?
You want to look at that?
Yeah.
Okay,
Can't
can
tell
we
what
have a
that
look
is.
at that?
Looks like a mishmash of maybe a few different colonies overlapping. There's a bubblegum coral too that I haven't seen yet. I thought I saw one that was covered with zoanthids earlier, but this might be something else.
That is quite a sponge.
I think even that's too much gain.
Can we briefly get the lasers on just to get a sense of scale, please?
It's a good shot
Wow.
of this in the still cam too.
All right, you want to push it on those corals there, please?
Thanks, Jake. You can turn the lasers off.
So this, the one on the left-hand side with that's red, it's almost certainly a bubblegum coral.
Yep.
Paragorgia. And we got our first bamboo coral.
Come a little wide, please.
Oh, yep.
Fell off my perch.
This is,
Like a heart print.
looks to be, actually, double check on that. Consult my bamboo coral cheat sheet.
Okay. While we're getting set up, Jake, I'm curious if we're feeling any more heave than normal on Argus, considering that
Go ahead
we're
and push on in there, please.
getting more swell coming towards us.
We've had a few
Catch up.
pretty big heaves, but nothing
Stephen,
crazy
when I
yet.
need to catch up pretty
Yeah, that's
soon.
all right. That's all right. Go
But,
ahead. I did
did you
some
get what you
images
needed,
of
or?
it.
Yeah, I
Oh,
think
good.
we're good.
Sure, thanks. A viewer has commented that after the last dive, the definition of very large sponge has changed.
Yeah, I think
Or
we
the
do need
standard.
to develop a size scale for our adjectives. Sarah was very kind and printed out a grain size cheat sheet for the lab, now that we've been collecting more sediments.
Oh, nice.
Definitely a lot of particles in the water. Some of them looking pink and spherical, but maybe that's just my imagination. Definitely coming from the southeast. We're driving right into that current.
Yeah, look at this current.
Looks like we're a little bit downslope. We can
Yeah, it does seem that we're kind of off of whatever.
Yeah, I can try to kind of come up it a bit with the next move.
Yeah, if we could maybe try to bump it so that we get up to the east a little bit.
Can do. Still some movement on this one. And,
Oh, great. I was beginning to think that the science chat wasn't working, but it's wonderful to see at least one of our shore-based scientists joining us. Welcome. We've been having some internet issues out here, so it's always a surprise. Lindsay, do we have a sense of how many people are joining us today, or is that something that's hard to find? With
I don't have access to that right now,
okay.
But I do know, because someone just informed me, that we have 418 viewers on YouTube.
Oh, thank you so much for letting us know, audience. That's great, and thank you for joining us for this exploration. Is it just me, Steve, or, you know, when we landed, there were a couple black corals in the view. We haven't seen as many as we've been moving, or
yeah, it could just be the saddle effect.
Yeah,
it's the saddle effect. Should pick up a little bit.
Folks are intrigued by your comment about stuff in the water on the last dive, Steve, and wondering how, first of all, how do you know that, or how can you tell when coral are gravid? And second, could we have slurped some of that out of the water column and to see if there was living things in it?
Yeah, we, so sometimes you can see corals when they have eggs.
Oh.
So, sometimes you can see through the body walls of the polyps that they have very yolky egg masses inside. It's sometimes possible, probably not with the ROV camera, to see larvae if they're brooders. That seems a bit more difficult. But usually you can see discoloration, like opaque yolky egg masses inside the polyps. That's, of course, no indication that they're ready to spawn soon, but it just means that they're in the process of accumulating eggs.
And that they have enough energy to.
Yes. But like we established several dives ago, there's no real...
Do you want to get a partial on that?
Good evidence. Yes, please.
Okay. Yep. Jess, if we have time, if we could get a closer look at that one.
Yeah, sure thing.
There's no real good evidence for areas like this where there is more sparse research about reproductive cycles for these types of deep water corals, and even then all corals are different on how they, what kinds of cycles they might reproduce on during. The second part of that question was about the larvae. How do we know?
Yeah, could we have slurped it?
Yeah. Slurp, no. I think the mesh size is much more coarse than what that could have been. The one thing we were discussing is to find a cloud potentially and just capture 2 or 3 Niskin bottles and filter the whole volume. And
go ahead, Jeff.
You know, potentially even in our 1 liter, though not extremely likely, if we got something like that and we were to sequence the DNA or to extract out the DNA, we would potentially notice a large number of reads from a certain
a little wide there.
Organism, and that could indicate that there's a whole larvae in there, for example. I think this is Acanthogorgia. Yeah, it's a lot easier to see from this angle on the plane here.
Okay.
So good observation.
All right. Thanks, Jess and Jeff.
Thank you.
Avoid.
Oh. Settle down there.
Sorry.
Some of these features look pillowy now.
Yeah,
But
we've seen a couple spots where it got more boulder-esque, but still a lot of what looked like sheet flows, relatively thin sheets too. Although now, given our experience the last few dives of maybe finding aggregated sand, I'm like second guessing myself.
Oh, it's okay. I'll get back over to you. Yeah, sorry.
Yeah, Argus is curving to the
To the
west.
south.
Yeah, more westward, south. We're changing to 135, but we haven't finished our 150 move yet. So
yeah, so the thing that I was looking at during the descent was the slope map we had pulled up on HiPAC, and I was a little concerned that the slope in this area might not be enough for really good flow to be induced, to speed over the top of this ridge. But it seems like it's good enough. I mean, there's not a lot of big things here, but there's a lot of small things that you can easily miss if you are not zoomed in partially or lower to the seafloor. So there are a lot of black corals. There's a lot of smaller sponges, some big sponges, but no like real big, tremendously huge corals like we saw on the previous dive.
Yeah, right, we haven't seen that yet. Although we're just now getting, Rennie, could you zoom out just slightly on HiPAC?
sure.
You know, we kind of started on this little tiny bit, but I think we're going to start getting steeper.
Right.
So.
No, yeah, I'm most certainly probably just in time for us to end our watch, as it always is. Fun stuff. Yeah, so there's some new stuff coming in here now. There's an Umbellapathes. I think we saw one of them just when we sat down.
There.
But yeah, good.
Exactly.
Good cluster of Chrysogorgia as well, Paragorgia, Hemicorallium.
Sorry, you guys wanted to get a quick sit down? Is that?
No, I was just pointing it out as he was saying it.
Right.
Yeah.
Making audio notes.
For our audience at home, if they're not coral experts, try to pinpoint what he's saying.
Yeah, I wonder if we could do text on text bubbles.
Yeah, I need like a cheat sheet of names that I could just be like, it's this one, it's that one.
Yeah, actually, we probably could, but it would take a lot of programming on that.
Yeah, no,
Pop-up video?
I could see that
But
going.
pop-up video can be fun on the Telestrator.
Fun facts in real time.
So have we spotted a bamboo coral yet, Steve?
Yeah, that one we
zoomed in on?
zoomed on a while back. That was behind the sponge. Was a bamboo coral.
Okay, but not like the whips that we've seen.
Not unbranched. Yeah, it was a nodal brancher. Potentially a J-clade, but I'm not certain about that. Could just as easily have been Isidella or something like that.
Again, one of these sponges that's just totally loaded with associated organisms.
Looks like Argus is feeling that 135 now.
Yeah.
Oh. Yeah, I don't know. We'll have to summon some sweets.
Yep.
Yeah.
Oh, what is this? Coming into view. One of our friendly fireworks.
Yep. Yeah, another Ridogorgia colony. There's some bigger stuff here. Yeah. I might want to take a closer look at that sparsely branching
Okay.
V-shaped
Yeah, up to the
colony
right.
in the background. Yeah.
Yeah, this one up here.
Roger that.
And yeah, we'll probably want to get some close-ups there.
Okay.
Since this is
How are we doing on our ship's move, Rennie?
We've got 10 meters left of that last move.
Okay.
This is really neat. If we could look at that left branch where it's, yeah, it kind of has those alternating. Yeah,
Sure.
that's a little weird. There's also something.
Yeah, I
Is
see
that
that.
another branch up there?
It kind of looks like a little proto branch.
Yeah. I'd like to look at the left side first.
Sure.
Kind of right in the zone that the branches are.
Go to push
branching.
on in there, please. Let's get there.
So any closer? This is kind of a, if anyone can find the node relative to the branch point, that would help. It's back here and super sharp.
Looks internodal.
It looks like there's a node right above it, almost. Oh, that might
Yeah,
just-
yeah, that is interesting. Yeah, I see that.
I see a node.
I see. Yeah, I think this.
There's
yeah,
a node right there.
Yeah, that's what I was
Yeah.
just
Oh,
pointing
is that
at.
what you're
I
pointing
think
at?
there's
Yeah.
also one just below the second branch.
Yeah.
There's a dust discoloration there. This
Like
is
in
interesting in that.
And there's one up here.
Yeah,
Right, you
the spur. Yeah.
You want to see the right branch before we have to go?
Yeah, please. Yeah,
Right.
if we have time to
Great shots.
quickly.
Lined
Yeah.
that up great.
So the one characteristic of these sparsely branching colonies
Do you
that
wanna
we've
look up
been
here,
instructed
Steve?
to look for. Yeah,
Okay.
if we have time.
Yeah,
That
we
was a
can
really
get
good
that.
Yeah,
zoom
we'll
we had. So if we look, we're trying to see if the node occurs just below the branch point. So technically on the internode, but also at just above that node point.
All right, Jeff, go ahead.
This one may be a little bit more difficult because it's kind of at a weird angle, but I think I see this dark discoloration just below the branch point.
Right there, yeah.
Yeah, which would indicate that this is one of the sparse branchers we've been keeping our eye out for.
Is this something we want to try to sample?
Yeah, it would actually be helpful to sample it.
Okay.
But I don't want to hold up the dive if we're.
Well, if Rennie, if you could put a mark here.
We're fine.
We are?
Yeah, it's pretty flat here.
Okay.
So actually, Argus could just end up on the other side of Hercules, and we could set up if you want.
All right. So would we like to try to
Yeah.
sample this?
Yeah, it would be great. I'm trying to think
Full
of
wide,
where the
please.
easiest place to sample is. We don't need a big piece. We just need maybe a small branch. I'll let the pilots in the front row think about which one is easier. But maybe one of the small secondary branches, a piece of that on the left side or the right side.
Yeah, so one of these smaller ones.
Yeah, left
Yeah.
side or right side, either.
Seems actually pretty tall up there. I think we're going to do like a fly up and grab type.
You want to do the sample the top?
Yeah, up to you guys, your call. Go ahead, Jake, and get the arm out there.
So just checking, you don't need a branching node. You just want
yeah, that would
snip
that would
of
be
it.
taking way too much. So I
Okay.
think just a smaller piece.
smaller piece.
Roger.
There's actually
Oh.
instructions right
Okay.
here about sampling for these.
All right.
Part of a power.
You gotta flip
PowerPoint
your jaws,
presentation.
rotate them. Otherwise, you'll
Yeah.
Yeah, smaller piece.
So maybe 10 centimeters, 15 centimeters
Gonna
maximum
hold it there for a sec. I'll come
would
down a
be
little bit.
ideal.
Yep.
I'm going to go for that one up top.
Where do we want to put this, Steve, once we collect it?
Depending
Can
on
you
the
get a little
size, actually, it could be slurpable.
Out there.
Okay.
Snip and slurp. Is that what you're?
Snip and slurp. Yeah, let's leave the boxes
Is
open.
this good enough, Steve, here?
I think they want to
Yeah.
snip and slurp, so we'll get a smaller sample.
Yeah.
Smaller, okay.
For snip and slurp smaller, yeah, that's that right there.
Right there?
It's fine. Yeah.
It might still be a little too large, guys.
Okay.
I'd
A little
go,
bit more?
yeah,
There
right
you go.
about there. That'd be good.
Sorry. Got
It's
it there?
like trying to hold onto a wet noodle.
All right.
I'll come a little forward there, Jake, if you want.
Okay.
So you have more scope.
All right. All right.
And if possible, before we do the
Awesome.
slurp, can we get a tight zoom on the closed polyps in the jaw?
Sure thing.
They're falling down back here.
Go ahead there, Jeff. All right, Jake. Nice.
There we go. That's exactly what we're looking for.
Awesome.
Very nice.
Very nice. Nice grab.
Okay, we can stow.
Roger. Yeah, hopefully this will fit to the slurp. We'll see. It's kind of large, guys, but
Yeah.
Okay.
so hopefully it doesn't clog.
It's pretty flexy, if
Gotcha. As long as it's
but
flexing.
not rigid.
Jake,
if
I'm
that
going to
makes
just
any
flush it
sense.
really quickly
Okay.
before you put it over there.
Roger that.
Like it won't.
Sorry, delta.
Did you want to take a look at the base where you can see the nodes better?
It's more about where the nodes are relative to the branches, the secondary branches, not so much the primary branches.
All right.
So that would be a primary branch point deep down in the colony.
Okay, section zeroed. We'll go to sample jar number.
So we can see in channel 3, or the lower left quad, the view
Number
of
one?
the slurp
Right.
chambers. It's kind of like a underwater vacuum. So we're gonna,
We got full jam on the section.
All
All
right.
you, Jake.
all right, so now you'll see
Follow you on the bubble
in the
here.
Herc cam, holding this up to the little nozzle in the front. That's the front of the
There you go.
suction. We're gonna try to suck that coral into that chamber.
Looks like we got some leftover sediment hash stuff.
Yeah, I see that, some little barnacle hash in there.
That's all right, yeah. So this would put it into clade D1, and it is identical to
rotating
the
your
clade
jaws a little
D1
bit.
that has been sent to us by your scientists ashore.
Nice. I get you better visuals.
If it turns out to be unruly, I guess we can go elsewhere, but try and give it a slurp first.
I gotta shoulder up, I think.
Yeah, you can shoulder up and then come in this way if you want.
Yeah.
Nice.
That'll be easier.
That'll be a lot better.
Yeah.
This is part of an ongoing project to identify from video if we can better identify
Looks
the
like you're in there.
branching
Go ahead
corals.
and gently let it go. Nice.
There it went in.
There it goes.
The branching bamboo corals from the unbranched bamboo corals.
Cross your fingers.
And
Yeah, can we shake the hose a little bit? See if...
There
Oh, there
it
it
is.
is.
There it is.
All right.
All right.
Yay.
So what's the sample
Good.
number there?
160.
160.
160, yeah.
All right. Bamboo
Nice.
coral.
Nice grab.
Snip.
Thank you. Good flying.
All right.
Okay, so Rennie, the ship is currently
We're
stationary.
Yeah, we're stationary. Argus is in front of us, but we'll just get caught up there, and then we can, let me zoom out and show you where we're at here. Argus is facing 135, so it looks like upslope is probably 150-ish. So we could do a 150 move.
Okay.
Head up towards that slope that we see there. Maybe
Yeah.
Maybe it's closer to 165, 160.
I like that
There's
plan.
one part.
Yeah. Can you face 160, Jake? And
Yep.
I'll just see where that slope lies.
I hear a magical sound coming from the front row. Yeah, really interesting pavements up here.
So we're looking at hot.
And again, the current seems to be coming at us from the southeast. What's that?
I was just mixing up ha'aheo and ho'oika
Oh, yeah.
seamount.
I think that's pretty straight on, yeah.
Yeah,
That's it.
I'll just go 155. Nah, 160.
Some viewers say they have the final dive blues.
Well, it won't be too long before Herc is in the water again and Argus is in the water again. The next expedition will start up again very quickly, just about a week.
Yeah, I think it's scheduled to launch December 8th. And then I believe the ROV dives would be beginning a few days after that,
Yeah, they're much closer.
weather permitting.
I think it's a day,
Yeah.
maybe a day and a half.
Yeah, it took us about a
Day.
day to get to the southernmost seamount when we mapped it on this particular cruise.
Water clear. It's hard to tell. The water clear is significantly different because the Argus camera is a bit different than what we had on earlier dives, but at least in Herc's view, it does seem to be more.
Part of it is with those lights, what I'm doing here is pulling the blacks down
Okay.
to try and keep it from being so murky. And if I just kind of zero that out, even without the lights.
That's true, yeah.
Yeah, it's seems to have more stuff in it than what we've seen previously. And it's hard to judge on the Argus camera because that's actually a different camera.
Yeah, exactly.
and I don't have the ability to change that picture at all. It's pretty much just what it is.
Okay.
So.
Jess, can you pan up to the left? I'm curious what this dark thing is.
I can't hear you so well.
Yeah, it's hard to
Oh,
hear.
I'm sorry. Can we
Up
get
to
a
the
look at
left?
whatever this dark material is?
Yeah, sure thing.
Vanta black holothurian.
A very large holothurian.
Is it just a
The
shadow of a rock?
Downside of a rock.
Yeah, okay.
So, hole to the center of the earth.
Yeah.
Yeah, exactly. Okay. I'm just seeing shadows. Thank you.
Sure thing.
Argus hasn't started to feel that move yet.
Edge.
We'll soon.
Is there a crab on that Walteria to the left? I
Let's take a look.
vaguely saw something that looked lanky like a crab.
Are we looking for this elusive white ghost crab?
No,
No, it was
I'm
red.
looking for any crabs at
It
this
looked
point.
like it was right in the middle there.
Oh,
Something there. Yeah, I couldn't tell.
go ahead and do a partial there. Like someone's
I
shag
like
rug.
it. Interesting how the
Maybe it's a brittle.
when Chris was on the watch up here, he was
Oh yeah, I think it's just two brittle stars.
yeah,
and a
not a
crinoid.
crab.
2 brittle stars and a trench coat.
Yes.
Thanks, Jess.
Sure thing.
That's a watch name, huh? 6 brittle stars and a trench coat.
Let's zoom in on those corals.
So this is like the last
I
watch
got
of this
chapped over
way.
Tasty corals.
When Chris was up here, he finally
You're gonna go partial on
taught
this
me
guy?
a word.
gonna
that-
jump.
Go, 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 go.
Do it.
Oh,
Oh.
no.
Don't do it. Don't do it.
Oh,
It's like-
don't you go.
I was like monkeys in a barrel, hanging on.
Wow.
And he stuck the landing.
Yeah.
Yeah, with only 4 arms.
Are these gastropods here we're seeing?
What do you see? Oh,
Yeah, kind of in the crevices there.
Yeah.
I don't know. We can take a look.
Yeah, let's take a look.
That's new in the scene.
All right, go ahead and push on there a bit, please.
Yeah, the Walteria sponges
Barnacles.
we've
Are they
been seeing.
barnacles?
Yeah.
Oh, yeah.
Barnacle.
Tiny crab.
So
Yeah, there's
that
a
would
tiny
make
crab
sense.
Oh yeah, there it
Yeah.
is.
Another one.
There's a squid lobster. Yeah, that would make sense. What we were seeing in the crusty flakes last night
Yeah.
actually resembles some of the contours of these
I agree.
Yeah. All right.
barnacles. So
Pull wide there, please.
revise that to barnacles, not bivalves, but it's a very, very interesting perspective how we had both reasonably heavy manganese coating as well as relatively fresh
Yeah.
calcium carbonate textures and colors. The Walteria sponges, yeah. Chris was up here for a brief period during a few watches ago, and yeah, he was saying those projections that come off of the Walteria, I have never known what to call them. Apparently they're called lateralia, which is a great word. But I was really impressed in the other first colony we saw that thought we saw the crab on, had them all flowing in the same direction.
Huh.
And I'm wondering what the function of that would be, because most of them have them coming out from all sides, but that one was really apparently swept.
Did you say lateralia?
Lateralia.
Oh man, that is good.
That is a good word.
Another one for your bingo card there, Rennie.
Yeah, lateralia. That's pretty easy to remember.
Yeah.
Ladder,
Stickopathes.
ladder rails.
Ladder rails.
Go ahead and push on their partial, please. That's great.
That one's pretty dark
Very
red.
red.
Yeah.
Very red.
Yeah, it looks like a bubblegum coral. Paragorgia.
Ah. All right, full wide there, please.
Falling off the map.
Yeah, sorry. I'll catch up.
Oh, you're fine. 25 meters to go with Argus. Ship's got another 7.
Reg.
I think I'll go the same way. It looks like it's fine.
So for the next cruise, those seamounts are just not mapped at all, right? Other than the one pass we did?
No, I believe they are mapped, if I'm not mistaken. I can't recall who did it. It was Falcor or Okeanos, but I believe that there is significant mapping data there. And we were filling in a gap. Well, I think half of that lower seamount was not mapped, and we were kind of adding a pass to it.
Let's see.
But this was a couple weeks ago, Steve. Can't be trusted.
Can't be trusted.
Seamounts, you know, when you start taking rocks off those seamounts, they just pop right back up again.
Yeah.
All the rocks that we've taken off the Pacific Plate.
Oh yeah, we never completed our weight estimate readjustment. How many pounds of rocks have we collected? I'm gonna go with 888.
I overheard one of the other scientists.
That's
I'm going
probably
like
going to be pretty close. for
1100.
Both cruises.
Oh, Jeff's going 1100.
1100.
1,000 on the
Okay.
nose.
1001.
I'll give it.
I was going to say Rennie spent his childhood, watching The Price is Right.
I did.
Yeah, so again, looking like kind of sheet flow like here.
Yeah.
Yeah. Really thin. So we're just past the 1950 meter mark.
I would say that the highest density and diversity we saw was actually in the
Right
place
where
we
we
touched
landed.
down. Yeah,
Yeah.
it hasn't been the same since. Did we put a target on bottom with a note about?
Yeah, I can double check, but yeah, I did. High coral and sponge diversity. Pretty much right at the star target. So.
Still similar here. You have some of the smaller species like the bathypathes, the stauropathes, the belopathes, but you're missing the primnoid fans, which I think
We're also
kind
not
of
seeing
unique.
those big sponges either.
Yeah.
Can I come up a little there, Jake?
Yep.
Thanks. It's a large dead Voltarian sponge.
Looks like there might have been one of those polyopogon sponges on our starboard side that we passed.
Oh yeah, just fading out of view in Argus
Yeah,
there.
exactly.
Oh, yeah. Is that a Victor? Oh, no. It's pink.
And I like the retro look of the SD, but this is much nicer. Felt like I was taking pictures of Argus with a Game Boy screen or something.
You're really going to have to measure that monster rock that was taken the last dive.
Okay.
Everybody wants to know.
Yeah, was there a weight for that one? That was
Not yet.
oh.
No, yeah, it used to be part of our standard practice way back in the day to weigh each rock, but we found that after you dry them, the weight changes somewhat depending on what the material is.
Interesting.
You know, with more porous rocks, like carbonates and stuff, holding more water and take longer to dry out. So I don't know why we stopped doing that. If there's a geological reason to not weigh the rocks when you first get them, maybe they weigh them at the repository. But yeah, it's also difficult, on a heaving ship. Your weight is kind of just a
scale.
Yeah, an estimate of a range of values.
Well, and what they really need is like a specific density, right?
Yep.
So just getting a weight of the rock, I don't think is very informative. Got to have a known volume and the weight. Where are you, fishies?
What are the
Brittany, do you mind zooming out on HYPACK a little bit?
Sure. How's that?
Yeah, that's great. I just want to see what we have coming ahead, what our chances are of getting back into a higher density area. Okay, thanks.
Sure.
Sorry, I might have interrupted you, Lindsay.
Oh, it's okay. I don't understand the question.
You know, from the look of the bathymetry, I would expect this particular stretch kind of from here to from the on bottom to where we are now to be more flat and it's pretty slopey.
Yeah, it seems like there's a lot of local variation. It's kind of undulating and hilly.
Some large.
But not really too bad. I mean, the Argus scanning sonar hasn't shown much relief. It's kind of like these local patches that Herc's been driving up and down.
Little knolls.
Can we zoom on this fan with the crinoid coming up? Small one.
Yeah, sure.
It's a big crinoid. Can we get the lasers on briefly? Thank you.
How big do you estimate it to be, Beth?
Well, those lasers are 10 centimeters apart, or 4 inches-ish. And so each one of the little arms of the crinoid is probably almost 15, 20 centimeters. Thanks, Jake. You can turn it off.
All right, go ahead there, Jeff.
This might be actually a paracalyptrophora based on density and size of the polyps. Color a little bit as a giveaway. I think most of the polyps are probably oriented downwards, and that's what I'm seeing. Parts of the branch tips when they retracted, places where they retracted.
Yep. Video, could we get a little bit more focus on the pink in the background?
On the coral in the background?
Yeah, on the coral in the background.
Yeah, I'll get you a little bit lined up there, Jeff. Oh, you're good.
Tilt up a little bit. Okay, great. That's a good shot. All right, good captures.
Okay.
Forward.
Looks like there's a large polyopogon off to our right there.
Ooh, very large.
Oh, yeah. A couple of them.
Yeah, a few in the Argus cam.
Kind of downslope.
Yeah, it's a big one.
Those are large, yeah.
Some big heaves here.
Yeah.
So those are
come forward for a minute, and then I'll
oriented kind of to the southwest, concave part.
Okay, go ahead and push past Bongo there. Thank you.
A few more meters on the ship move. Argus has about 20 meters left.
Reg.
Put lasers on that sponge.
Lasers?
Yeah, lasers on that big one.
Lasers.
Ooh, big one.
Yeah.
Could almost drive Herc inside that cave.